Hi, everybody. Welcome to Micro Seminar. I'm Lizzie Wilbanks. I'm one of the hosts and organizers of Micro Seminar. And we are really excited to have with us today Dr. Jeff Marlow. Jeff was a lab mate of mine in the Orphan Lab at Caltech. He graduated with his PhD in 2015 with a PhD in geobiology. And since then, he's gone on to work with Pete Gerges at Harvard and has been doing lots of exciting things there as a postdoc. So Jeff, I'm going to turn this over to you and let you take it away.
Sounds good. Thank you very much. And it's great to be with you all. This is a new format for me. Just talking with Lizzie that this is an interesting way to give a seminar. So we'll see how it goes. Feel free to reach out with any questions as we go. Afterward, I'm pretty easy to find. And we'll go from there. So yes, as Lizzie mentioned, I have been thinking kind of about these ideas in a geobiological sense about how methane and microbial communities on the seafloor really influence global distributions of methane and sort of the flux of these energy-rich molecules throughout the biosphere and the geosphere. This started as a big part of my PhD and has kind of continued as a pretty fruitful side project over the intervening years. So the data I'm going to be showing you today is a mix of kind of the first foray into this system of authigenic carbonates on the seafloor at Methane Seeps, and a continuation of that work to see what the limits of that system could be. So for some context, I know I'm kind of preaching to the choir, but microbes are really essential components of our planet. They control elemental cycles and the composition of the atmosphere in really important ways that dictate the habitability of various systems for the rest of us. So figuring out how these microbial communities work is really important to see our planet as it is and also as it was throughout geologic time. So with this in mind, my focus is really on culture-independent, activity-based studies of microbial communities. The first two questions that any environmental microbiologist will ask are, who's there and what are they doing? And those questions in turn kind of unlock a range of other questions, like how they are doing that and how these activities really impact Earth systems. It's also important to realize that we're dealing with a very, very wide range of spatial scales. From the planetary scale, the questions about sort of how greenhouse gases are distributed throughout the atmosphere, that's the biggest scale that we can think about, all the way down to the molecular scale of how different proteoforms could be grabbing onto substrates. That's 17 different orders of magnitude. So we need to be pretty clear about what exactly we're trying to ask and how we're going about answering it. Based on the scale that we're dealing with, our methods will vary substantially. And more specifically, I'm going to be talking about these methane metabolizing microbial communities, which is a really fascinating and important system, kind of epitomized by this photo here on the back, which is the Iberian Peninsula at night. And it really shows two aspects of the system that fascinate me. One is the use of electricity, the idea that methane is an important hydrocarbon component of natural gas and really fuels a lot of our use of energy. And the idea that we can think about how natural systems, natural methane metabolizing communities could be harnessed to facilitate this energy use is an interesting question. I won't go into it too much today, but it's another thing we've been working on. And secondly, that thin rind of the atmosphere, viewed from a global orbital perspective, is really just shows how tenuous that atmosphere is and how any changes to it can have really big repercussions for what's happening on the surface. So to put that in more perspective, most of the world's methane is locked up in hydrates, these ice cages of methane that can result in flammable ice. So if we think about the overall amount of methane in the atmosphere, based on this tiny square, the overall amount of methane locked up in hydrates takes up the rest of this slide. And within that small slice in the atmosphere, a pretty small proportion of that is coming from marine sources. And these really differing volumetric numbers are telling us two things. First is that this hydrate reservoir is really stable. To allow for such a wide range of amounts of methane between these two reservoirs, there isn't much net flux going on in between. But the second part is that any small changes to this decent equilibrium we've got now could have really big repercussions. Methane is a very strong greenhouse gas, about 25 times stronger than CO2 on a 100-year timescale. So any amount of methane that's getting from hydrates in the atmosphere could have pretty important implications. But on the seafloor, it's a very different story, where methane percolating out of the seafloor at areas known as methane seeps form the basis for rich oases of life. Per unit area, these are equivalent to some of the most dense and productive habitats on the surface, like the Amazon rainforest. And here we see some of that methane hydrate ice sort of outcropping at the surface and fueling this rich biosphere that is manifested on the macro scale by mussels, clams, then further trophic levels on up. And methane seeps are pretty pervasive too. This is a map from 2014, which is already hopelessly outdated, showing that we see seeps at a range of geologic contexts. We'll get more into that a little bit later. But really, we're just limited by where we're looking. And these things are, in fact, everywhere. The first step when looking into what could be happening just below the surface at these methane seeps kind of came from geochemical studies, when looking below the sediment layers around methane seeps, or even just in any marine sediments, this sulfate methane transition zone seemed to be a pretty diagnostic feature. Spots where sulfate from the seawater goes down and intersects with a zone of methane increasing. That happened kind of initially in this observation in the North Sea, then was also found to be occurring in Namibia, off the coast of Alaska, and off the coast of California. And this suggested that there was a process that was consuming both of these constituents, the sulfate and the methane. And the smoking gun for this process of anaerobic methane oxidation, which indeed does consume both of those reactants, came with fish nanosims investigations that showed that there are archaea and sulfate-reducing bacteria that link up in these consortia, and that when you feed them isotopically enriched methane, it does in fact go into their biomass. So this is tying function with identity and showing that there are these anaerobic methanotrophic archaea and sulfate-reducing bacteria that are accounting for this consumption of methane in anaerobic environments. And further studies kind of broke down this relationship as shown here, where the anemone archaea, here shown in this fish image as a red dye, are consuming methane, producing bicarbonate and electron equivalents, which are then transferred to the sulfate-reducing bacterial partner here in green. And that latter half reaction with the sulfate reducer is much more energetically attractive, and that kind of drives the entire process. The estimated amount of free energy available from this at standard conditions is about 30 kilojoules per mole, which is low, but it's not sort of beyond what is believed to be the energetic limit of life, where as measured through symbiotic partnerships, that could be negative 13 kilojoules per mole, or modeled as a biological energy quantum, it's about 20 kilojoules per mole. So it's more exergonic than that, which is encouraging. And one of the other main aspects of this system, especially at seafloor seeps, is that the bicarbonate links up with calcium ions in the seawater and precipitates calcium carbonate. This makes huge mounds of carbonate rock, as is shown here, with a range of side-scan sonar data from different systems. And across all of these systems, from Costa Rica to Hydrate Ridge to the Chilean subduction zone, the vast majority of methane perfused volume is carbonate rock. The thin drape of sediment is really just a meter or two, usually on the top, while the rock itself can account for hundreds of meters vertically of volume. And this suggests that if there is anything happening inside the rock, that could be really important on a global scale. This image is just showing that, again, these mounds are kind of everywhere. Seeps are everywhere that we look, at least along certain geological areas. This was from an expedition off of Costa Rica, which found a mound every 4 kilometers on average. Those are the red dots, and each one was about 1 kilometer wide and several hundred meters tall. So there's a lot of carbonate rock out there. And my main question when entering this field was what was happening inside the rocks. A lot of that initial characterization of the system came from the sediment. A lot of the rates and overall understanding of the volumes involved came from the sediments. But again, because there's so much carbonate rock out there, much of which is being percolated through by methane-rich fluids at Methane Seeps, any amount of biological activity in the rocks could have a really big impact. So this first started with a couple of expeditions to Hydrate Ridge, and this is one of the best studied methane seeps out there. You can see some refuse from previous years of study there up to the left, but it's really just a very productive system with a lot of bubbling methane, and you can also see some of that surface expression of active microbial communities in the white mats and some of those clam beds as well, which are pretty indicative of a lot of activity. So we collected some rock, mostly expressed at the surface, and brought it back to the lab to see exactly what was happening. This is a cartoon to kind of show the types of systems we were dealing with. So we've got these active sites where we know that methane is sort of percolating through all of that volume. And within that, we've got our sediments, which are that's been studied relatively well in the past. Active nodules, which are these small kind of protolithic chunks of carbonate. And then we have the rocks themselves. The porous carbonates would have these really pretty big conduits and massive sort of denser chunks of carbonate, usually farther down. There's also the off-seep area, or the low seep activity zone. We did not see any surface expression of activity here, so there were no clam beds, no microbial mats, but we can't really rule out that methane was being produced and perhaps fully consumed at depth. So we're looking at two main variables here. One is the substrate, the sediment, nodule, or carbonate, and the other is the degree of seepage activity, from low seep activity to these very active locations. First question, who's there? So we took a look under a microscope and found, again, these kinds of aggregates that we typically associate with AOM and methane seeps. At active sediments, we found something like this, about 5 or 6 microns in across. Active nodules with a general DNA stain, slightly larger. And at these active porous carbonates, some were even larger than that. And taking this into account, the overall size of aggregates, the number of aggregates, and the packing density of them, there was in fact 41% more biomass tied up in aggregates in the carbonate rocks than in the sediments. So this is a pretty good indication that there's a whole biosphere in there. But then in terms of what they were doing, that was the next question. Are they still active? Are they just kind of remnant forms of previous aggregates that were taken up through the sediment? We wanted to approach this in a range of ways to think about what kind of metabolic activity was really happening. So to think about this, we need to back up a little bit and consider the different types of metabolic activity when we say some of these terms. So I'm going to briefly walk you through the one way to think about this. And there are a number of different terms that we'll use going forward to specify exactly what type of metabolic activity we mean. So the first thing to think about is methane activation. This is where methane is being broken open by that first enzyme in the process, MCR, and leading to a range of downstream products. This is kind of agnostic of what happens after that initial cracking open of methane, which is a very stable molecule. So that's methane activation. Methane oxidation means that it's gone all the way to the most oxidized state, bicarbonate. Methanotrophy usually indicates that we're moving some of that carbon from methane into biomass. This is a relatively small proportion of the overall methane within these aggregates usually. Autotrophy accounts for the vast majority of the biomass. That's from bicarbonate. And then net methane consumption is really anything downstream of that activation. It's also important to realize that this enzymatic system can go both ways, and that trace methanogenesis has been shown to occur where you're taking some of those intermediates, pushing it back through the MCR enzyme, and actually making methane. This is also just half of this symbiotic relationship, and electrons being moved to the sulfate reducing partner leads to the other half of that equation. So these are the range of different types of activities we're talking about, and I'm going to walk you through a few different things that we looked at to hone in on some of these more specifically. The first was looking at biomass and methanotrophy with nanosims. This is nanoscale secondary ion mass spectrometry, which is a pretty cool way of sputtering away layers of biomass, taking up those secondary ions in a mass spectrometer to see what their masses are. And you can distinguish between carbon and nitrogen and other isotopes. We focused on those two to see, kind of to produce a spatial map of the isotopic distribution. And this can be linked up with fluorescence in situ hybridization to see exactly who you're looking at under the microscope. So this is one of the samples that we set up to take a look at this. You'll notice this was a 27-month incubation. These guys grow very, very slowly, so you got to be patient. And these were set up in gas-tight mylar bags and left with their isotopically enriched components of ammonium and methane. 27 months later, we cracked it open, and here's what we saw. Each row in this figure is showing a different aggregate of ANME and SRB, and each column is telling us a different way of looking at it. So on the far left, we have the cardfish showing us the phylogenetic distributions, with ANME in red, sulfate reducers in green. In the central column, we have the distribution of N15 and atom percent, and on the right, we have the C13. So this is most visible with the nitrogen. Those numbers are in the mid to high 80s in terms of the atomic percent of nitrogen, that is N15. That means that we had at least 3 doublings during these 27 months. And the carbon values were also enriched. Our control aggregates showed up around 0.5 atomic percent C13. So we're seeing substantially more than that in all of these aggregates. The next thought was to look at methane oxidation, this sort of catabolic side of this. And we could do that in a couple of ways. One was by looking at the overall amount of C14 radio tracer as it goes from methane all the way to the fully oxidized state. And another was this new system of monodeuteраted methane that we set up to look at the catabolic intermediates and sort of focus in on methane activation. So the C14 and CH3D, those are looking at different elements of methane, carbon or hydrogen. So they're going to tell us slightly different things. And when we tested this system out, we found something really remarkable. The first was that the ratio between those two systems, so the CH3D measuring kind of methane activation and how much of that hydrogen is going into the water, and the C14, which is showing how much of that carbon from methane is going all the way to CO2. These ratios were extremely consistent. It was right about 2 for pretty much all of these systems. And we're still trying to figure out exactly why it's 2, but the fact that it is so consistent, both across different substrate types, from the sediments to the carbonates, and across different levels of seepage activity, from these low activity locations to the active ones. This tells us that this is a pretty good proxy for measuring full methane oxidation, that this ratio of 2 is pretty reliable across a range of different communities to look at. So that's kind of about the systematics of how we're looking at methane oxidation rates. But another kind of more scientific and system-based answer we had from this study was that indeed catabolically methane was being oxidized at all of these locations. Again, within the carbonates for sure, but also at some of these low activity sites, both the sediment and the carbonates. And that was kind of unexpected to see that they remained poised to take up methane when placed in a methane replete situation back in the lab. All right, so far we have kind of done this initial characterization of the authigenic carbonates at seafloor seeps. We've looked at who's there. We've done a little bit of triangulation to figure out what they're doing with these different methods. And the next questions we're looking at are how they're doing it, and how these endolithic communities inside the rocks could be impacting Earth systems. 2 initial kind of side products that came out of this first effort. One, published by my colleague Andrew Thurber, showed that there are these worms inside the rocks that are eating archaea. So the lipids within the worms, only a few of them were not attributable to host biochemistry. Those are the ones here that I'm pointing out with the arrows. And they really could only have come from archaeal members. So these are archivory, archaea-eating worms, suggesting that inside these rocks, the primary producer, ANME SRB consortia, are really supporting a full ecosystem with various trophic levels. Another realization we had was that this is an intriguing form of endolithic life. There were kind of these 3 previously established forms of endoliths, either these euendoliths boring down into the rock, chasmoendoliths, or cryptoendoliths that are kind of in these remnant systems. But by precipitating carbonate that they are inhabiting, this is an interesting kind of new form of autoendoliths, self-endolithic organisms, which as you're forming rock around yourself, it leads to a lot of questions of whether or not this is a long-term good idea, or if things are kind of regulated as this strangulation may or may not start happening. But it also brings into question how we interpret seeps from the ancient past. There are several dozen known outcroppings of ancient seeps on land, and a lot of times we can look into those and see in these white arrows, these are kind of pointing to organic rich inclusions. The traditional interpretation is that this was the sort of accumulation and consolidation and compaction of sediments, but it could be that they were rocks all along and they were actively metabolizing through that entire lithification process. All right, so we have at this point shown that this happens one place. We've shown that it's occurring at Hydrate Ridge, that organisms inside the rocks are metabolically active. But the next question was how pervasive this could be. So this is where the kind of long-term side project samples of opportunity comes in. Over the last 3 years, we've been collecting some of these rocks and doing long-term incubations to see if this is a common phenomenon. So the next set of samples came from the Gulf of Mexico. This is a hydrocarbon-rich basin. It's a not an tectonically active location, so the vast majority of methane seeps, and especially those that are studied, most substantially are on the sort of west coast of North and South America, places where subduction is happening. but indeed there are seeps elsewhere. So Gulf of Mexico is one of those locations. And we picked up some rocks, and upon bringing them to the shipboard lab, we found that they were already seeping natural oil, which was really cool to see. You can see that in some of these dark spots on the left, and this sort of pool of black goo here in the sample on the right. And when we incubated them over the course of about a year, these are the kinds of methane oxidation rates we were seeing. I'm going to be building on this plot as we look at different samples, so this will start to look familiar, but you can see that the filled-in symbols are for rocks, the open symbols are for sediments, just to kind of compare what was happening in both types of substrates. And for comparison, that carbonate from Hydrate Ridge, the most active one, would plot about here. And the vast majority of previously published studies from methane seeps, from continental shelves and slopes, would kind of be in this zone that I'm highlighting here. So already we're kind of looking at the upper level of activity, especially within a couple of these sediments from Gulf of Mexico, but also a couple of the rocks as well. The next stop was these recently characterized seeps off the coast of New England. This is a passive margin. There's not really any subduction happening. So this was traditionally thought to be an unlikely spot to find kind of hydrocarbon seepage. But as you can see here, there is kind of a rich biosphere with these white microbial mats oxidizing sulfide, one of the byproducts of anaerobic methane oxidation, and higher trophic levels here with various fish and clams and mussels. So this was a series of 2 canyons off of New England that we were able to collect some samples. And here's where those data plot, here in orange. I'm showing 4 sets of rock samples, 2 from an active site and 2 from a seemingly inactive site. And everything plots relatively close. So regardless of whether these rock samples came from a location exhibiting signs of a lot of seepage or chemosynthetic activity or not, the communities appear to retain the ability to consume quite a bit of methane. Next stop was the Guaymas Basin. This is a heavily sedimented hydrothermal system in the Gulf of California. Again, an intriguing and different geological setting. And these are the data we got from there. So there's 1 rock sample that's really consuming methane at very high rates, and a few at lower rates, and some not really at all. There are a number of sort of energy sources and fluid conduits coming out at the Guaymas Basin. So there's a distinct range of geochemical environments available, some of which are really driven by hydrocarbons, some of which are not. So this is consistent with that idea. And our final system is at a spot called Point Dume off the coast of California. And here we found these really bizarre matte-encrusted chimney-like structures. They were often anywhere between about 10 centimeters and a meter tall, forming this really bizarre yellow brick road of carbonate chimneys. Just gonna zoom in on a few of them because they're so crazy and cool to look at. We collected a couple of these structures, brought them back to the lab, and here's what we saw. So over the course of more than 100 days, we see that methane oxidation rates are extremely high. Everything I showed you before was below this white dotted line. So we're seeing that these Point Dume chimney samples are about 1 to 2 orders of magnitude higher than some of those previous samples. And already those were at the higher end of rates of methane oxidation seen previously. So this was cool. We wanted to put it into context of previously published data. And to do that, we need to think about the methane concentration at which we were doing our experiments. So ours were all done 2 atmospheres saturated with methane. That's about 1.1 millimolar in the bottle. And here I'm plotting a lot of different previously published studies. And on the x-axis, we've got the methane concentration at which those different studies were done. This is obviously important because if you have more methane or at higher concentrations, you would probably expect a higher rate, even for the same kind of biological community and innate methane oxidizing potential. We can back calculate what some of these higher methane concentration incubations would have been using Michaelis-Menten kinetics. And when we do that, we can draw the lines back to where they would be at 1.1 millimolar, and this is what we see. So even some of these highest values at Black Sea and Hydrate Ridge fall below a couple of our samples at Point Dume. We also wanted to see what things would look like at higher pressure. That's one of the key environmental variables that is often not considered when we do kind of lab-based experiments. And we used the equivalent of about 750 meters depth, which is approximately where the Point Dume chimneys are. And across all of our samples, we exposed them to these pressures and found that, for the most part, things increased quite a bit. Here are these lighter blue samples on the right, showing what happens at high pressure and saturated conditions. Everything I showed you previously, again, was below this dotted line. And these green dots are all of the previously published data before we back calculated based on methane concentration. So again, we're seeing that a couple or one sample in particular is the highest sort of raw methane oxidation rate, regardless of methane concentration that we've seen so far. Okay, so the obvious next question is what's going on, especially in these rocks and in the Point Dume chimneys. Why are rates so high, or what variables could help account for different rates that we see? We took a range of approaches here, from kind of physicochemical parameters to biological and kinetic and thermodynamic parameters. I'm going to go through a few of them and highlight the ones that we seem to think probably matter a little bit more than others. First, we looked at the type of mineralogy. So here I'm showing the 22 samples we had, and you see the rate of methane oxidation averaged based on the replicates we had and over those longer timescales, sort of sorted out by different geological settings as well. We have the Gulf of Mexico hydrocarbon basin, the New England Seeps from two different submarine canyons at the passive margin, Guaymas Basin north and south at a hydrothermal rift basin, and at Point Dume and a nearby the Palos Verdes area along the California coast. So those are the different sorts of geological settings we're dealing with. These are the rates of methane oxidation. These are the sorts of mineral phases we saw when we did some X-ray diffraction analyses. They're the major constituents, perhaps unsurprisingly, were carbonates, a lot of aragonites, calcites with various magnesium, calcium ratios. The thing that is perhaps most interesting or surprising or relevant here is that our highest rate of methane oxidation came from this Point Dume chimney, and the interior was about 80% pyrite. So when we scraped off the kind of outer millimeter or two of those chimneys, it was almost exclusively pyrite inside. Some of these other samples, rocks 2 and 3 from Point Dume, they had trace levels of pyrite as well. So we'll get back to what that could mean a little bit later. So we also did some 16S analyses of all of these samples and looked at the alpha diversity. There are 3 different approaches to looking at alpha diversity here in the different rows, and the different columns show those 4 different geological settings that we're dealing with. The heat map gives us a sense of how quickly they were oxidizing methane. So really fast chimney-like structures here on the far right had moderate to low alpha diversity. Overall, there's not really a clear trend here linking rate to diversity. So we also looked at beta diversity, how the different communities kind of shape up relative to each other. So these are some NMDS plots looking at 3 different ways of presenting this sort of information. And based on this amount of discrepancy that's attributable to these two axes that we're looking at, we see that things sort of sort out by location. So we could perhaps convince ourselves that some of these Gulf of Mexico samples, especially over here, kind of plot together. The Atlantic margin ones tend to plot together pretty well. Some of the other ones are a little bit more mixed. But in terms of rates, things are really not too clear. So the size of the circles here are indicating how quickly methane oxidation is happening. Bigger circles mean faster rates. And some of those highest samples are actually pretty close to some of the lowest rate samples. So this beta diversity, the way that the different community structures correspond to each other as a whole, probably not a primary determinant of rate. So another way to look at this whole thing is through kind of the calculations of how we model rates of methane oxidation. So this can be done by sort of looking at the maximum rate when all reaction centers are filled and methane is replete, and that's sort of tempered by kinetic and thermodynamic terms that we can take into account. So we're going to start with the kinetic aspects. The main variables here being the concentrations or activities of methane and sulfate, the two reactants in this AOM system, as well as their KMs. And this is typical Michaelis-Menten kinetics, where the KM shows you kind of how strongly the biological community is latching onto and metabolizing the reactants. So lower KM means that is a more specific relationship, and higher is the opposite. We did a study using different methane concentrations with those point Dume carbonates. That's going to be these filled-in squared data points, and co-localized sediment from the same spot, point Dume. Those are the circles. And if we just look at that circle plot, so this is the same kind of plot here that is typical for Michaelis-Menten kinetics, where we vary the substrate concentration here on the x-axis and expect an increase in the reaction rate until we kind of reach saturation and reach that Vmax. That's what we're doing here. And with the circles, with the sediment, as we increase the methane concentration, we see that it's a very consistent relationship to what we'd expect. It asymptotes out here at a Vmax, but that the carbonate rock is doing something pretty different. For some of these lower concentration samples or data points, things are pretty consistent, but then it really just keeps going up, or perhaps enters a different phase space where we see that there doesn't seem to be any Vmax, at least below 129 millimolar methane. And that's really interesting. We can think about why that could be happening in a moment. And then the other aspect of this is the thermodynamic side. So here we're looking at the Gibbs energy available, temperature, some of these other factors, and taken all together, we were kind of trying to schematically sketch out all of the different variables that could play a role kinetically and thermodynamically to influence the rates that we're seeing around different structures. So this is a very busy plot. I'm not going to walk you through all of it, but the boxed parameters are the things that we think matter. So for example, the rate at which methane is taken up into the cell, the number of MCR, the sort of rate limiting step of methane oxidation, how many copies of the enzyme are available per cell, how quickly electrons are going between the two symbiotic partners, etc. There are all these different things that probably matter based on our sort of modeling of the thermodynamics and the kinetics. There are three of these that we're going to look at real quick. One is the activity of methane. That is similar to the concentration of methane. So what is the localized availability of methane that cells are seeing in their microenvironment? Second is this kind of substrate electrical conductivity idea that electrons need to flow between these two partners. And if you had a conductive surface on which both partners are adhered, that could be helpful. And finally, the overall cell abundance. More cells, more reaction centers, more methane being taken up and metabolized. So we're going to start with that this methane activity idea. The localized concentration of methane is not the same as the bulk concentration that you add into your incubations. And in fact, there could be a localized adsorption of methane to the carbonate rocks. So the thought was that perhaps, as sort of shown schematically here, there's a lot of methane very close to the carbonate rocks due to various chemical reactions. This has been shown for organic-rich shales in the past, where the total organic carbon concentration is directly proportional to the methane absorption capacity, so that as there's more total organic carbon in the structures, there's more methane adsorbed at surface layers to these structures. Making methane adsorption measurements is something that oil and gas companies do, which means, or cause or effect, who's to say? It's very expensive. We did not go down that route, but by using this total organic carbon percentage as a proxy, we can use a couple of these previously established best fit lines to see how that could affect our different systems. These data are still being collected, but it looks like there's at least a 10 to 20% amount of that rate difference could be attributed to the methane absorption capacity through the total organic carbon. So this could matter. It's probably not what's driving things, but it seems to be an important role, at least in terms of concentrating that methane locally and allowing for faster rates at the rocks. The second aspect is following up on that XRD measurement of the pyrite inside. So this is a sort of cross-section view up one of those chimneys. This is that interior, this is the outside, and you can see these kind of cool layers that are forming. And the interior is dominantly, at least of a couple of these, was dominantly pyrite. Under the SEM, we saw these really cool pictures of framboidal pyrites, which sort of confirmed that those were present. This is just a pretty picture of some aragonite needles. Because when you're scooting around on an SEM, why not take glorious photographs when you can? And so this is still, we need to follow up more on the exact conductivity parameters of these chimneys, and that's still sort of ongoing. But if pyrite is a dominant constituent of some of these structures, that could allow for electrons to flow much faster through them, and kind of relax that requirement that the anomie and sulfate reducers need to be in direct physical contact. If they're merely in direct physical contact with a conductive surface, then that could allow for kind of higher throughput of these metabolisms. And then finally, and perhaps most straightforwardly, we looked at the amount of biomass within these structures. And you can see that this is our point Dume chimney that had the highest rate we've seen. It also had the highest amount of biomass. The difference in biomass cell abundance scales pretty consistently with rate, but it does not account for all of that higher rate. It's anywhere between about 15 and 44%, depending on which pairwise comparison you're making. And that's also assuming a consistent per cell rate of methane consumption. But it seems like there's something going on in the rocks that holds on to or retains or facilitates higher cell densities. And that alone is enough to account for a pretty decent proportion of the high rates that we see. We also looked briefly at what sorts of metabolisms could be happening based on the 16S libraries we made. There were, as you'd expect, a lot of anomies, the anaerobic methanotrophic archaea. There were a lot of sulfate reducers. We were also looking for kind of the next layer of these metabolisms. Were there methanogens or were there sulfide oxidizers? These would consume end products of AOM, which could allow for higher throughput of those reactions if the products no longer are building up. So that's what I'm trying to plot here, where on the x-axis we have a log scale of rate of AOM. So again, these are our point Dume structures. And then on the y-axis we have relative abundance of these different types of microbial constituents based on metabolism, expected metabolisms. So the ANMEs are in these filled red and green, sulfite reducers in the purple X's, sulfite oxidizers in these teal figures. The point being that sulfide oxidizers, and actually none of these really tracked with overall methane oxidation rates. ANME2s were showed the best relationship, where they were very abundant, among the most abundant lineages in the high rate systems. But the sulfide oxidation story and the methanogen story, which I didn't plot here, do not really show any relation to rate. So the other cool thing we were able to do is to look at what's happening, how these constituents are arranged in their native habitats. So we took a chunk of these point Dume carbonate rocks and looked at the reflected light. And this is a stitched image across several fields of view. You can see the scale bar here, 50 microns down at the bottom. And this is a reflected channel. So the white is showing us the surface of the rock. The black is showing us the pore spaces. And then on top of this, we can view with fish the archaeal members in red and sulfate reducers in green. And this is not concentrated. This is just microbes natively as they're occurring. And it's an amazingly dense habitat. And we also see really cool mixes of sort of shell type aggregates here, more mixed systems over here, sort of monospecific layers lining some of the pore spaces. Here are some archaea, here are some sulfate reducers. So by looking at these communities as they really occur in place and in relation to each other in pore spaces, and ultimately to minerals, this could be a really powerful way of seeing what's happening inside the rocks. So that's kind of where things stand. Hopefully we've convinced you that these methane oxidizing communities inside the carbonate rocks are important. They seem to be pervasive. And based on some of the rates we're seeing, I think that they really warrant a reconsideration of the overall amount and distribution of methane consumption in the seafloor. So I'll leave you with this quick thought: the idea that anything you ever read about methane oxidation is going to cite this number of how much methane is being consumed globally, and a lot of that's attributable to AOM, but this does not account for what's happening inside the rocks. And I think we've shown that there are three main things that really are going to be changing this number. It's a decade in the past at this point, and a lot of things have changed or come to light to show us that this number is probably a large underestimate. First, again, is that we're seeing there's a lot of activity in the carbonate rocks. Carbonates account for the vast majority of the volume at Methane Seeps. Second is that we need to consider the in situ methane concentrations. Most of the numbers are from these sorts of experiments come from atmospheric pressures or concentrations of methane that don't really reflect what's happening on the seafloor. And then finally, new seeps are being discovered all the time. We're really just limited by where we're looking. And again, that probably just means that this system is more active all over the place than we ever imagined. And with that, I would like to thank the two labs that have really helped out, or that I've been a part of while doing this work, Caltech with Victoria and here at Harvard with Pete, as well as a number of great collaborators and some of our funding sources. So thanks again for tuning in, and I'd be happy to take any questions.
Thanks, Jeff. That was great.
Of course.
Exciting work. So I'm going to be collating questions here. So if you've been watching remotely and you have questions for us, you can tweet us your questions and I will ask them here to Jeff on the air. So we will be watching out for that. And in the meantime, I'm going to get Jeff warmed up with one initial question while you all tap away typing us your questions. So Jeff, the finding that you found about the pyrite in the Point Dume chimneys is super cool. So you mentioned the suggestion that it might be that they're electrically conductive, and so they're dumping their electrons out onto that surface. What do you think are some of the limitations in terms of, you know, what the aggregates or the individual organisms would need in order to do that, and the length of sort of the little pili they'd need to stretch out, or things like that? Like, what do you think are some of the limitations to that possibility?
Sure. Yeah. So that's a good question. One is that ideally they would, need some way to be directly conductive, but perhaps not too encapsulated, in the sense that, you want to be able to tune that reaction and to control it. So to be able to kind of reach out and grab or distribute electrons and then cease that when you need to stop it. Because it is going to be an electron, sort of a common electron marketplace, and it's going to be tuned similarly for all members so that as the redox potential changes, that flux could come in and out. So you want to kind of be able to control it. Yeah, we haven't gotten any good images yet of potential pili or extensions or anything like that. So at this point, it is kind of the next hypothesis that we haven't really zoned in on quite yet. But I'd also say that it could be a part of this kind of product drawdown system idea as well, where it's not, we showed that there weren't really many sulfide oxidizers who would get rid of that sulfide, but if it's precipitating, that effectively is doing the same thing. So it could help kind of in a thermodynamic sense as well.
Yeah, that's what I was wondering about, the pyrite sort of being that sink. Or
that's right. Yeah, though interestingly, we don't, we saw it mostly with that one sample and not some of the others. So it could be sufficient, but not necessary, I guess. It could be one of a variety of ways to increase the rate, depending on exactly where you are and what the geochemistry is.
Yeah. All right, let's see if we have any questions here. It looks like everybody might have a case of the Monday mornings in terms of class participation.
No problem.
Other questions, but let's check one more time, and then we will go offline. Do you have anything? Why don't you tell us about some of the things you're most excited, or projects that that you're excited about, and sort of the up and coming things that you're working on?
Sure. So that last image that I showed of kind of looking at the microbial communities in place on the rock surface is kind of the seed of the idea of the next project that we're working on. So this is a Moore Foundation project with Roland Hotzenpickler's group and Pete's group, where we're looking at sediments starting in Sippawisset in Cape Cod and trying to preserve the microscale arrangements of microbial communities as they are, naturally. And it's remarkable to me that most of the work that the environmental microbiology community does is kind of bulk or through concentration. Like we'll get things on filters and look at how they are arranged then. But to really conserve things in place and look at how different organisms relate to each other, to different minerals, and how those activity rates could change is really cool. So I've been playing around with different types of resin to freeze them in place and section. And I think there's some exciting downstream microscopy we can do from there to answer some of these questions of how heterogeneous they are and how activity really changes with horizon, with mineralogy, and with their neighbors.
Awesome. Well, tell me when you get that working. I want to try it.
Sounds good. We'll touch base in 5 years.
That's awesome. All right. Well, I don't see any questions here, but if any of you come up with questions watching this later, because this will all be saved in our archive, you can, Jeff is on Twitter. He's JJ_Marlow, or you can tweet at Microseminar, and we will pass your questions along. So thanks so much, Jeff. It's been great having you.
Of course. Thank you so much.